Let's see what is osmosis and osmotic pressure here from the chapter solutions, class 12. Yes. When we look at what is osmosis here, see, if when I've taken, I'm conducting this particular experiment where I'm taking a solvent here in this, and I'm putting a setup like that. This apparatus will contain the more concentrated solution, maybe a sugar solution or a salt solution, and this is just a water. This is a solvent, right? This is more concentrated solutions, means I'm adding a solute to a solvent, right? This is what more concentrated solution means. See, if I if I put the membrane here, we can see this as semi-permeable membrane, SPM. What does a semi-permeable membrane mean? The semi-permeable membrane is the one which allows the flow of only solvent molecules through it and does not allow the solute or any particles to move through it, or is called a semi-permeable membrane. This semi-permeable membrane we occur in our, naturally in our blood cells or living cells. All the blood cells are, have this membrane called as this semi-permeable membrane. This is a cell membrane, in other words, which allows the flow of only solvent molecules and not the solute. So this is permeable to what? Only the solvent molecules. Such a membrane, it's called a semi-permeable membrane. And this is actually the pig's bladder, or it can be of animal or vegetable origin. So this is what a pig's bladder, or it can be the parchment paper also. Or it can be synthetic, it is synthetic, such as cellophane. So all these act as semi-permeable membranes. That means which will allow the flow of only certain substances flow through it, that is solvent in this case. Okay, so when I'm fixing a semi-permeable membrane which allows the movement of only solvent molecules. So what do you think? So the solvent moves, or the water particles, it's a solvent in this case, solvent molecules will move from lower concentrated solution. This is lower concentrated solution, right? Because this has only pure solvent, low concentration. Here it is of higher concentration, right? Higher concentration solution, because here we are solute particles we are adding to it. And so it becomes concentrated because of the presence of solute is in it. So the solvent which is moving from these this particular beaker into this apparatus like this, right? Here the concentration of solvent is less. So since the semi-permeable membrane allows the movements of only solvent molecules, the solvent molecules will move from outside to the inside of this funnel, right? Till when it will move? Till equilibrium is reached. Till equilibrium is reached. That means it will, till the concentrations becomes equal inside and outside, till then the solvent molecules will move inside. Okay, right. Because here solvent molecules are more, here solvent molecules are less. So this, since the semi-permeable membrane allows the movement of only solvent particles and not the solute, so solute particles cannot come out. Only solvent will move in until the concentrations becomes equal, right? So this is what is osmosis. So when a semi-permeable membrane is placed in between a solvent and a solution, the solvent molecules flow through the membrane from pure solvent to the solution. This movement of the solvent molecules from the low concentrated solution to the higher concentrated solution is nothing but the osmosis, right? Yeah. Now, how can we stop this osmosis taking place? Just, yes, if we can prevent this osmosis, that means the movement of the solvent molecules, by just applying some pressure on this. So if you put some pressure on this, the flow will stop. And this pressure is nothing but the osmotic pressure. So what is this pressure? This is nothing but the osmotic pressure. Right, let's look at this. What is osmotic pressure now? Yeah, this is what it is. It's the solution which is containing what? This is a solution containing solvent molecules and solute particles. right? These are solute, these are solvent molecules. So here pure solvent, only solvent molecules. So this is a semi-permeable membrane. So the solvent will move from the pure side where there are more number of solvent molecules to the where there is lesser number of solvent molecules. Otherwise, this is of low concentration of solute. This is high concentration of solution. Right? This is what is called osmosis, the movement of the solvent molecules from the solution of lower concentration to the solution of higher concentration is nothing but the osmosis. Right. So I said that what is our osmotic pressure? So osmotic pressure, in order to stop this osmosis, right, is nothing but the pressure that must be applied to stop the movement of the fluid or to stop the movement of the solvent or to stop the, in other words, to stop the osmosis which is occurring through this semi-permeable membrane. Right. Here I'm taking this solvent and solution in a separate experiment. See, if I'm applying some pressure on this solution, actually, if there is this is a semi-permeable membrane in between these two. So here it's solvent and solution. So the movement will take place from lower concentration to higher concentrated solution, right? So this will solvent molecules will move in this direction. But if I stop this process of osmosis by putting some pressure on it, this is called as pi, nothing but the pi. So this pi is called as what? Osmotic pressure. This pi is called as what? Osmotic pressure, right? Pi is the osmotic pressure. We can stop this osmosis which is occurring here, right? Now, here this osmotic pressure is directly proportional to the molarity, not the molality, is directly proportional to the molarity, which can be given by this equation, where C is called the molarity, R is the gas constant, and the T is the temperature at which this is experiment is carried out. Okay, this is what is happening, right? Let's do let's see a few calculations here. Yes, I said the pi, the osmotic pressure, in order to stop the osmosis, this pi is nothing but in order to stop the osmosis is directly proportional to what? The molarity. C is the molarity here, R is the gas constant, T is the temperature, right? This molarity is nothing but is equal to number of moles of solute by what? Volume of the solution in liters. Right? This is what is molarity. Now, this is nothing but number of moles of the solute. Well, if you take it as W2 grams of this is dissolved in 1000 grams of the solvent, right? I mean, 1000 grams of the 1000 ml of the solution. This is taken as a volume, right? So its molar mass is M2. W2 is the molecular weight of the, W2 is the, W2 is nothing but the weight of the W2 grams of solute is dissolved in the solvent, which is having the molar mass of M2. This number of moles is equal to W2 by M2. Volume of the solution in liters. So this one, if we take it in ml, this one is the formula, right? If we substitute this, pi is equal to C is what? W2 by M2 into 1000 into T. Right? RT. Right? If we avoid, if you don't want to put this 1000, no problem. So this is for V is in liters. So W2 into R2 by M2 into V. Done. Or M2 is equal to, from this equation, W2 R2 by pi into V. So if you know all the variables here, we can calculate the molar mass of what? Solute. This is M2 is what? Molar mass of solute. So using this atmospheric osmotic pressure, we are able to find out the molar mass of the different solutes or different substances. W2, if the known weight or of this is dissolved in the known volume of the solvent and yeah, Everything else we know here. Yes, R is the gas constant. So M2 can be found out, right? We can calculate the molar mass of the solute. Yes. Then what are the applications of this osmosis? Where is this osmosis and osmotic pressure being used, right? Measurement of this osmotic pressure. Yeah, this is for what? This is for the, yeah. This one is for what? This osmotic pressure we are measuring because we know to when we need to know the molar mass of the solute, right? This is the first one. This is the first application. So this is a colligative property, right? This osmotic pressure is what? And osmotic pressure. This osmotic pressure is a colligative property. Colligative property is nothing but the property which depends on the number of the solute particles dissolved in the solvent and not on the nature of the solute, right? That is what is colligative property. Since this is a colligative property, when we are finding, with the help of the other colligative properties like elevation in the boiling point or depression in the freezing point, all those experiments were carried out at different temperatures and pressure. But this one we are maintaining at the room temperature and pressure, right? This is one, room temperature and pressure we are doing this. So this is one advantage of calculating the osmotic pressure in order to find out the molar masses. And one more thing, this is used to find out the molar masses of what? The molar masses of biomolecules actually. So these biomolecules like proteins, carbohydrates, or macromolecules, anything, right? So actually these macromolecules, all these are denatured if the temperature is high. This can be studied, or the molar masses of these can be found out only when we are doing this at a lower temperatures. That is why this has an advantage over the other methods, because we are calculating all this at room temperature and pressure. And one more thing, this is directly proportional to the molarity, but all other methods are proportional to the molality there. And the magnitudes or the numbers we are getting are large here, even for the very dilute solutions. So these are very accurate, but the other methods, very small values, not very accurate. So we're dealing with the big numbers here, the values will be very accurate. And yes, this biomolecules, these are the applications of, right, osmosis or osmotic pressure. Then what are isotonic solutions? The isotonic solutions are the solutions which are having the same osmotic pressure, right? So the solutions which are having the same osmotic pressure are nothing but the isotonic solutions, which means that if I put a semi-permeable membrane in between these two isotonic solutions, the osmosis does not occur, right? Does not occur. So the osmotic pressure which is associated with the fluid inside the cells is 0.9% mass by volume. So if I want to give the intravenous injections, so this will have the, 0.9% mass by volume sodium chloride solution actually. So the saline which we are injecting intravenously is nothing but 0.9% MBB. So it's the same concentration we are giving, so that no osmosis occur and there is no effect on the cells, on the blood cells, right? Now, so this one, this osmotic pressure is actually used where in order to, in pickling, where preservatives, right? In making pickles, we are adding lots of salts. In sugars and jellies, we are adding lots of sugars here, right? Jellies, jams, all this. So this will do what? They will not help, allow the bacterium to grow. That means they will kill the bacterium, which is, coming and attacking on this. If they are, bacterium will come on this pickles, what will do? Because lots of salts we are putting. These salts are what? Highly concentrated. So what will they do? They will allow the osmosis to take place. So what will happen? The water or this is of low concentration, the pickles are high concentration because of salts which are present in it. So osmosis will occur and the water is, taken out from this bacterium. As a result, the bacterium is killed. the shrink and killed, right? This is what I mean. This method is applied for storing or preservatives for pickles, sugars, and all this. Okay, and if we put some what do you get, wilted flowers into the fresh water, also they will again regain, they will become healthy again. How this is happening? This is a process. This is based on again osmosis only, right? These are the applications of osmosis and osmotic pressure. Now, what is reverse osmosis? What is reverse osmosis? Yes, this reverse osmosis is this is a reverse of osmosis. That's it. In osmosis, what is happening? If I take a fresh water here, salt water here, put a semi-permeable membrane in between, how does the osmosis will take place? From the solution of lower concentration to the solution of higher concentration, right? So the solvent molecules will move from the solution of lower concentration, that means fresh water, to salt water. This is what is osmosis. But reverse osmosis is what? If I put a piston, if not allowing the fresh water to come into this, so I'm putting some pressure on this, what will happen? So pressure which is more than that of the osmotic pressure, just not to stop the osmosis, but the pressure which I'm applying is more than this osmotic pressure, which means I will allow the water to flow from this salt water to the fresh water, right? This is a reverse of osmosis. Now that is what is reverse osmosis, right? Because I'm applying pressure. How much of pressure? Not just equal to the osmotic pressure. If it is equal to the osmotic pressure, it will not allow the osmosis to take place. But I'm putting pressure which is more than osmotic pressure. So the water from this is forced into the fresh water now. Right, this is what is reverse osmosis. So where this is applied, this reverse osmosis is used for purification of water, right? So water purification, this is generally used. That is reverse osmosis, right? Some pressure is applied. So this is used for desalination of seawater, so that it is used for drinking purposes. Okay, yes, for domestic purposes, seawater. So what is a membrane? This is the membrane which is used is a semi-permeable membrane which is made of cellulose acetate, right? So this is what is osmosis and osmotic pressure and its applications. Yeah. If you like it, please do like, share, and subscribe my channel. And if you want any topic to be discussed, please comment in the comment section. And yes, trainings are also given from Asia Chem Academy for all the grades and for all the boards. Yes, please interested, please see the description box below and the details are given there.